or the cells of body, and that's why regulation is also necessary. Once C5b, so as we have seen that different pathways of this complement, that is classical pathway, alternative pathway, and mannose binding lectin pathway. In all this pathway, C5b, that is the molecule generated, and this C5b attracts C6, 7, 8, and finally number of molecule of C9 attach over here, and they form a larger complex known as a membrane attack complex, and this membrane attack complex bind on the surface of nearby cells, especially the target cell, that is C5b678 complex, and along with this C9 molecules, number of C9 molecules bind and create a pore that is having sufficient pore size of 70 to 100 angstrom, and this is sufficient to kill the cell, target cell, because as the pores, number of pores are created in the cell, especially the cell membrane, ions and small molecule diffuses through the channel of membrane attack complex, and it is very much difficult for cell to maintain the integrity because the free movement of ion is there in the cell from the pore created by membrane attack complex. Thus the cell cannot maintain its integrity and eventually results in the lysis of the cell. So this component, especially C9, when it doesn't bind with the C5, B, 6, 7 and 8 complex that bind on the cytoplasmic membrane, that is sufficient to create a small pore that is 10 angstrom, and that is sufficient to lyse non-nucleated cell, for example RBCs, but nucleated cells cannot be lysed. For lysis of nucleated cells, binding of C9, that is prerequisite. So once C9 bind and polymerize over here, that is with C5b, 6, 7 and 8, that creates a large pore that is having 70 to 100 angstrom pore size, and that is sufficient to kill even the nucleated cells. And you can see in the next slide, the intact bacterial cell that is acted upon by membrane attack complex. As you can see over here, the projections coming out from the microorganism from the bacterial cell, that is due to activity of membrane attack complex, and that creates number of pores in the microorganism, and this result in the formation of pores, and ions freely coming out from the cell eventually carry out lysis of the cell. Once C5b molecule is there, this C5b molecule when bind with C6, 7 and 8, then conformation changes are also found in that, because the cytoplasmic membrane, as you know, that is consisting of bilayer, lipid bilayer, and the conformation changes take place in the C5b678 molecule that exposes hydrophobic site of this complex, and this once the hydrophobic site is exposed, that interact with the hydrophobic molecule of the cytoplasmic membrane and anchor itself deep inside the cytoplasmic membrane. So as and when required, conformation changes are taken place in the membrane attack complex, especially C5b678 molecule, and then it anchors inside the cell, ultimately form a membrane attack complex. Apart from this, other biological consequences of complement activations are there, that is opsonization, immune clearance of the antigen antibody complexes, opsonization and phagocytosis. As you can see it over here, IgG molecule bind on the bacterial cell cover with complement, that is seen over here in triangular form, and antibody, specific IgG antibody bind over there the bacterial cell, especially on the bacterial cell that is covered with number of this complement protein, and also it helps in the activation of complement. Similarly, phagocytosis, on the phagocytic cell number of receptors are there, as you can see it over here. That is CR1 complement receptor 1 is also there, FC receptor is also there on the phagocytic cell. Phagocytic cells are those they try to engulf the microorganism, the foreign organism, and destroy it. If foreign organism is when it is opsonized by complement system and specific antibody bind over there, it has in the phagocytosis of the bacterial cell, ultimately result in killing of the foreign microorganism, whatever it may be. And as you can see it over here, with help of complement receptor, this bacterial cell can be engulfed and later on that is killed. Clearance of immune complexes, when large number of antigens, circulatory antigens are there, and antibodies, specific antibodies are found in the serum, in the blood, antigen antibody complexes are generated, and these complexes carry out activation of complement system, and once the complement system gets activated, it results in clearance of these circulatory complexes, that is antigen antibody complexes, and that is very much necessary. As you can see in this slide, that erythrocyte RBCs, they are having receptor, that is CR1 complement receptor 1. So antigen antibody complex is there, you can see it over here, the brown color antigen antibody bound to that, and on the FC portion of antibody complement is there. Specific receptors are there on the erythrocyte, that is complement CR1 receptor. This antigen antibody complex bind on the surface of erythrocyte, and this is taken into the liver or spleen, where clearance of this coated RBCs with antigen antibody that are already cleared. Again, alternative is there, antigen antibody complex, when the antigen antibody complex bind with C1 receptors are also there found on the phagocytic cell, and this phagocytic cell bind complement with complement, that is already bound to antigen antibody complex, and ultimately result in clearance of these complexes from the serum or the body. Now regulation, now we will switch over to regulation of the complement system. It is again very much necessary to regulate the activity of the complement system, because if it remains active for longer period of time, then along with the foreign organism, host cells are also damaged, and that's why many elements of the complement system are also capable of attacking host cells. So regulatory mechanisms have evolved to restrict complement activity to targets. Regulation of complement system, that is carried out at different stages, as we have discussed in the pathways, different pathways of complement system. The complexes generated at different level, for example C3 convertase and C5 convertase. So these active complexes are controlled either before assembly or after assembly. We'll start with when the assembly has not taken place, that is before assembly of convertase activity. One of the protein that is known as C1 inhibitor, C1INH, that binds to C1R2S2, causing dissociation from C1Q. So once it dissociates from the C1Q, as you can see it over here, a specific black color molecule, that is a C1 inhibitor C1INH, that inhibit C1R2S2, that releases R2 and S2 from C1q, and ultimately no activation of complement system, especially for the cleavage of C4 and C2 is prevented, because for cleavage of the C4 and C2 the activity of C1 S2, that is crucial one. So C1 R2 S2 that releases from the C1q molecule with help of C1 inhibitor. Another one protein that association of C1 C4b and C2a, that is blocked by binding of a specific protein known as C4b binding protein, that is C4b BPP, that complement receptor type 1 or membrane cofactor protein, that is MCP. As you can see it over here, once the cleavage of C4 is there into C4a and C4b and C2 into C2a and C2b, C4b and C2a they bind with each other and form C3 convertase. Before binding of C4b to C2a, a specific protein known as C4b binding protein or CR1, that is complement receptor type 1, or MCP, that is membrane cofactor protein, that bind with C4b molecule and won't allow C4b to bind with C2a. And once C4b2a complex is not there, the formation of C3 convertase that is prevented. And once this factor bind, another factor 1 is there that cleaves out C4b into C4c and D. In alternative pathway, CR1 MCP or cofactor H prevent binding of C3b and factor B also. So C3b, that is a portion, especially component of alternative pathway, the same thing is there happen over here also, and prevention of complex C4b binding protein, CR2 of factor H, and DAF, that is decay accelerating factor. Once the assembly has taken place, then DAF, decay accelerating protein, that carry out dissociation of this protein, and no activation of C5 convertase is there. Another protein is there found, that is known as S protein, that prevents insertion of C5b67 membranatic complex into the membrane. If C5b67 already formed, then S protein bind over there and won't allow C9 molecules to bind over there, C8 and 9 molecules. Homologous restriction factor and membrane inhibitor of the reactive lysis, that is CD59, that binds to C8 and preventing assembly of poly C9. As you can see it over here, C5b678, when it binds the factor HRF of MIRL, that prevent binding of C9, and once C9 not bind over that, a very small pore is created, and that is again self regulated, or the assembly of the cell, that again pore, whatever the pore is created, that is again cleared. Then number of these regulatory proteins are there. If these proteins are not there, deficiency of the complement system is there, that leads to different kind of diseases. For example, deficiency in C1q, R and S, that leads to disease systemic lupus erythematosus, that is generalized autoimmune disease, and deficiency into C4 and C2 leads to glomerulonephritis, vasculitis, staphylococcal infections, a factor D deficiency or properdin deficiency, that leads to infection by certain bacterial agent, and deficiency in C3, that leads to recurrent severe bacterial infections, immune complex diseases, and so on. Membranatic complex deficiency, that leads to infection with meningococcal, gonococcal infections, and complementary regulatory protein, that is C1 inhibitor is not there, that is responsible for hereditary angioedema. Thank you very much.
Thank you. Thank you once again to this, for this opportunity, to all KCG team, BISAC team, and my principal, and the main, that is SANDHAN team. Thank you very much.
Again, thank you to the